I welcome all the registered students in the SWAYAM course of Genetic Engineering and its recent advancement. In previous lecture, you have been gone through the topic farms animals. The topic that we will cover in today's lecture is genome editing. Myself, Dr. Kush Kumar Nayak, Assistant Professor, Amity Institute of Biotechnology, Amity University, Chhattisgarh. Today we will cover following topics, that is introduction to genome editing, techniques for genome editing, applications of genome editing. We will see ethical consideration in genome editing. We will also check the regulation and challenges associated with genome editing and also check the future direction in genome editing. So, let us start with the introduction part. The characteristics of all living organisms are determined by their genetic material and their interaction with the environment. An organism complete set of genetic materials is called as its genome, which in all plant, animal and microbes is made of long nucleotide of DNA. The genome contains all the genetic information needed to build that organism and allow it grow and develop. Within the genome, there are regions called genes. These genes are made up by DNA sequence of nucleotide. These genes can carry instruction for making protein, which in turn give the organisms its characteristics or traits. While every cell in an organism will have essentially the same genome, the difference between cells are determined by how and when different set of genes are turned on or off. Occasionally, changes to DNA in cell can occur that create a new and different version of a gene, which can then be carried by that organism's offspring. These changes are known as mutations and means different individuals can carry different version of that particular gene, which can result in difference in trait within population. For example, for individual eye color. So, genome editing is a method that can let scientists change the DNA of many organisms, including plant, bacteria and animals. Editing DNA can lead to changes in physical traits like eye color and disease risk. Scientists use different technology to do this. Gene editing, the ability to make highly specific changes in the DNA sequence of a living organism, essentially customizing its genetic makeup. Gene editing is performed using enzymes, particularly nucleases enzyme that have been engineered to target a specific DNA sequence, where they introduce cut into the DNA strand, enabling the removal of existing DNA and the insertion of replacement DNA fragment. Key among gene editing technologies is a molecular tool which is known as CRISPR-Cas9. CRISPR-Cas9 function with precision, allowing researchers to remove and insert DNA in the desired location. Genome editing is a revolutionary scientific technique that enables scientists to make precise change to DNA of living organisms. This powerful tool has the potential to profoundly transform various fields, including healthcare, agriculture and environmental conservation. By manipulating the genetic code, genome editing allows us to correct genetic disorder, enhance crop yields and develop innovative way of tackling diseases. Genome editing has revolutionized the field of genetics and biotechnology by allowing scientists to precisely modify genetic information, which has various applications in research, medicine, agriculture and more. It has opened up new possibilities for research and has the potential to transform various fields that include medicine, agriculture and environmental conservation. However, the ethical implication and potential risk associated with genome editing raise important questions, necessitating careful consideration and thoughtful regulation. In this lecture, we will delve into the detail of genome editing, exploring its technique, applications, benefits and see its ethical concern that surround it. The techniques used in genome editing. In recent years, the emergence of highly versatile genome editing technologies has provided investigation with the ability to rapidly and economically introduce sequence specific modification into genome of a broad spectrum of cell types and organisms. The core technology now most commonly used to facilitate genome editing are cluster regularly interspaced short palindromic repeats associated protein that are known as CRISPR-Cas9. Second, transcription activated like effector nucleases known as TALENs, zinc finger nucleases known as ZFNs, and homing endonucleases or mega nucleases. Let us discuss one by one how it performs the whole process. The first genome editing technology were developed in the late 1900 and more recently a new genome editing tool that is known as CRISPR invented in 2009 has made it easier than ever to edit a DNA. CRISPR is a simpler, faster, cheaper and more accurate than older genome editing methods. Many scientists who perform genome editing now use CRISPR. Genome editing techniques like CRISPR-Cas9 have provided researchers with a powerful tool to study genes and their functions in a more targeted and efficient manner. However, the utilization of this technology also raises ethical questions and concern, making this topic of a great importance for university studies. Genome editing, a groundbreaking technique in the field of biology, has generated significant attention for its ability to modify the genetic makeup of a living organism. With the advent of advanced genetic engineering tools, scientists have gained unprecedented control over the DNA sequences of organisms, allowing them to insert, delete or replace genes with remarkable precision. This remarkable capability has opened up vistas of possibilities in various domains, ranging from curing genetic disease to enhancing crop resilience. CRISPR-Cas9 uses RNA DNA binding rather than protein DNA binding to guide nuclease activity, which simplifies the design and enable applications to a broad range of target sequences. CRISPR-Cas9 was derived from the adaptive immune system of a bacteria. The acronym CRISPR referred to the clustered, regularly interspaced short palindromic repeats, which are found in most bacterial genomes. Between the short palindromic repeat, there are stretches of sequence clearly derived from the genome of bacterial pathogens. Older spacers are found at the distal end of the cluster and newer spacers representing more recently encountered pathogens are found near the proximal end of the cluster. Transcription of a CRISPR region result in the production of a small guide RNA that include hairpin formation from the palindromic repeat linked to sequence derived from the spacers, allowing each to attach to its corresponding target. The RNA DNA heteroduplex form then bind to a nuclease called Cas9 and direct it to catalyze the cleavage of double stranded DNA at a position near the junction of the target specific sequence and the palindromic repeats in the guide RNA. Because RNA DNA heteroduplex are stable and because designing an RNA sequence that binds specifically to a unique target DNA sequence requires only knowledge of the Watson-Crick base pairing system. The CRISPR-Cas9 system was preferable to the fusion protein design required for using ZFN and TALENs. A further technical advance that come in 2015 when Zhang and colleagues reported the application of CPF1 protein rather than Cas9. As the nucleases paired with CRISPR to achieve gene editing, CPF1 is a microbial nuclease that offer potential advantage over Cas9, including requiring only one CRISPR guide, RNA form specificity and making staggered double stranded DNA cuts. The altered nuclease property gave potential greater control over the insertion of replacement DNA sequence than was possible with Cas9. At least in some circumstances, researchers suspect that the bacteria house other genome editing protein as well, the evolutionary diversity of which could prove valuable in further refining the precision and versatility of genome editing technologies. So, let us understand how this CRISPR work actually. CRISPR leverages a natural defense mechanism of a bacteria to cut DNA at a specific location. Specifically, when bacteria are attacked by a virus, they recorded a section of the virus DNA in their own DNA, bookending it with a repeating sequence called cluster regularly interspaced palindromic repeat. Storing part of the virus's genetic code allow the bacteria to remember it. When the same type of virus attack again, the bacteria use a specific CRISPR associated protein number 9, which is Cas9, to cut the virus's DNA, destroying the viruses. In the laboratory, scientists use the same methodology with the CRISPR-Cas9 system to identify and cut a specific DNA sequence. They do this by creating an RNA sequence that matches the DNA they want to edit. Then, using these RNA sequences to guide the Cas9 protein to that location, scientists can efficiently reprogram Cas9 to cut DNA at any desired location. Once the CRISPR-Cas9 cut the specific DNA that has been targeted, scientists use other technology to add, delete or modify the DNA. As shown in figure, CRISPR-Cas9 system allows scientists to make targeted change to an organism's DNA. Researchers are already building on a CRISPR to create new gene editing systems. For example, a system known as a prime editing build on the knowledge of CRISPR-Cas9 to directly exchange one piece of a DNA with another, which is known as base editing. Since its development in 2012, CRISPR has generated significant excitement because of its faster, cheaper, more accurate and more efficient than prior methods. As a result, interest has grown rapidly and CRISPR is now used extensively in thousands of laboratories worldwide. Further, CRISPR based treatment for certain diseases including sickle cell anemia and some type of cancers are currently in clinical trials. However, CRISPR has not yet been approved by the US Food and Drug Administration for medical uses, nor has FDA issued regulations on food safety for crop edited with CRISPR. The another technique which is used in genome editing is TALENs, that is transcription activator like effector nucleases. Transcription activated like effector nucleotide, that is TALE, has recently emerged as a revolutionary genome editing tools in many different organisms and cell types. The site specific chromosomal double strand break introduced by TALENs significantly increases the efficiency of genomic modification. The modular nature of the TALE central repeat domain enable researchers to tailor DNA recognition specificity with ease and target essentially any desired DNA sequence. Engineered nucleases enable the targeted alteration of any DNA sequence in a wide range of cell and organisms. The newly developed transcription activated like effector nuclease comprise a non-specific DNA cleaving nuclease fused to a DNA binding domain that can be easily engineered so that TALENs can target essentially any sequence. The capability to quickly and efficiently alter any gene sequence using TALENs promises to have profound impact on biological research and to yield potential therapeutic strategies for genetic diseases. Let us understand how does TALENs work. Targeted genome editing is a broadly applicable approach for efficiently modifying essentially any sequence of interest in a living cells or organisms. This technology relies on the use of engineered nucleases, that is artificial protein composed of a customizable sequence specific DNA binding domain fused to a nucleus that cleave DNA into non-specific manner. These targetable nucleases are used to induce double strand breaks, that is known as a DSBs, into specific DNA sites, which are then repaired by mechanism that can be exploited to create sequence alterations at the cleavage site. Nucleus mediated genome editing enables genetic studies that were previously difficult or impossible to perform. This technology might also provide therapeutic avenues for genetic disorders, including monogenetic diseases such as sickle cell anemia or cystic fibrosis. Figure representing symmetric diagram of the talons, where tail repeats are shown as a colored cylinder with a final carboxy terminal truncated half repeats. Letters inside each repeat represent the two hypervariable residues. Tail derived amino and carboxy terminal domains required for DNA binding activity are shown as longer blue and gray cylinders respectively. The non-specific nucleases domain from the FOC1 endonucleases is shown as a larger orange cylinder. Talons bind and cleave as a dimer on a target DNA site. Note that tail derived amino and carboxy terminal domains flanking the repeats may make some contact to the DNA. Cleavage by the FOC1 domain occurs in the spacer sequence that lies between the two regions of DNA bound by the two talon monomers. The schematic diagram of tail derived DNA binding domain, the amino acid sequence of a single tail repeat is expanded below with the two hypervariable residues highlighted in orange and bold text. Tail derived DNA binding domain align with its target DNA sequence. Note the matching of repeat domain to single bases in the largest site according to tail code. Also note the presence of 5' thymine preceding the first base bound by the tail repeats. The third technique used for genome editing is ZFN, which is Zinger, which is zinc finger nucleases. Zinc finger nucleases have been used to modify endogenous genes in a wide range of organisms and cell types. Several types of genomic alteration can be introduced with ZFNs, including point mutation, deletions, inversions, duplications and translocations, thus providing researchers with unprecedented tool to perform genetic manipulation. Furthermore, ZFNs can potentially be used for therapeutic purpose. Cis2 and His2 zinc finger are DNA binding domains that each recognize approximately 3 base pairs of DNA. Alteration of a small number of residues in or near an alpha helix within this domain can lead to change in its DNA binding specificity. Engineered zinc finger can be joined together into more extended array capable of recognizing longer DNA sequence. However, to obtain protein with high affinity and specificities, context dependent effect between and among finger in an arrow should be considered. In addition, various publicly available methods for engineering zinc finger proteins have been described. Modular assembly of individual pre-selected zinc finger domains provides a simple and rapid method for creating customized array, but the success rate of these strategies has been reported to be low. Additional methods that explicitly account for context dependent effect among zinc finger in an array include oligomerized pool engineering known as OPEN, context dependent assembly known as CODA, and bacterial 1 hybrid known as B1H. The bacterial 1 hybrid selection based system. A large number of zinc finger arrays engineered using these various methods have been fused to a non-specific nuclease domain from the type 2S FOC1 restriction enzyme to create zinc finger nucleases. The FOC1 nuclease functions as a dimer, and therefore, two zinc finger array must be designed for each target site. Engineered zinc finger array have also been fused to transcriptional regulatory domain to create artificial transcription factor capable of altering the expression of endogenous genes. Let us try to understand how does ZFN work. Each subunit of zinc finger nucleases is composed of the three domains. First, a non-sequence specific cleavage domain at C terminal mediated by fork1 nuclease. Second, a DNA binding zinc finger domain, that is cis2 his2 at the N terminal, essentially for its specificity. And third, a peptide linker that connect the zinc finger domain with the nuclease. The restriction enzyme fork1 induce a DNA double strand break as a catalytic dimer. Thus, for DNA cleavage to occur, two zinc finger subunit must bind to the gene target sequence in the opposite orientation, leading to fork1 dimerization. In addition to its nuclease activity, effect of engineered zinc finger on DNA extend to artificial transcription factor and methylases. ZFN is composed of a 3 finger binding domain and fork1 nuclease cleavage domain that are connected by a linker. Each finger binds to 3 base pair. Site specific double stranded breaks can be induced by ZFN at endogenous loci. The repair process lead to small gene deletion or insertion in non-homologous end joining pathway. Corrected gene or transgene can be integrated into the mutant gene by an alternative mechanisms, homology directed repair, while co-expressing ZFN and donor DNA. Let us understand the various application of a genome editing. It can be applicable in a human health and medicine. Treatment of genetic disease, genome editing provide potential therapeutic approaches for genetic disorder by correcting or modifying diseases causing gene mutations. Cancer research, genome editing allows scientists to create better models of cancer, study its mechanism and develop more efficient treatment. In infectious disease, genome editing can be used to modify disease vector or enhance the body's immune response to combat infectious disease. Development of a new diagnostic test, targeted medicines and other treatment. By pairing CRISPR with other Cas protein that cut the target DNA or RNA and then cut other nucleus to produce a visual signal. Scientists can identify when viral components are present. Scientists are using this technology to develop diagnostics tests that may rapidly identify diseases such as COVID-19. CRISPR-Cas9 is binding tested for the possible treatment for certain diseases including sickle cell anemia and some type of cancer. CRISPR may also be used to help control certain diseases by altering the trait of insects or other organisms that can transmit disease. For example, scientists use CRISPR to make mosquito more resistant to the parasite that cause malaria. The second field where it used is agriculture and food security. Genetic improvement of crops, genome editing enables the modification of crop characteristics to increase yield, enhance nutritional value and improve resistance to pests and diseases. Livestock improvement, genome editing can be used to introduce beneficial traits in livestock such as disease resistance or increased productivity. Stronger, more disease resistant crop. CRISPR may improve agriculture product by increasing nutrient content, disease resistance and survivability in adverse weather and soil conditions. For example, when researchers deleted a small portion of specific cucumber gene, the plants were less susceptible to viruses known to damage the plant and cause significant crop losses. The third area where it applicable is conservation and environmental applications. Preservation of endangered species, genome editing can help save endangered species by addressing genetic issues and improving their chance of survival. Ecological restoration, genome editing might allow for the modification of invasive species or the introduction of genes that promote biodiversity and ecological balance. Let us focus on the certain challenges and ethical consideration related to the genome editing. Regulatory framework. Various countries have different policies and regulation regarding the use of genome editing techniques, especially in human applications. Intellectual properties. The development and the use of genome editing technologies raise challenges related to the intellectual property rights and patent disputes. Public acceptance. There is a need for public education and awareness about genome editing to foster acceptance and address social concern. Safety concern. There could be unintended consequences of using CRISPR. For example, it could target an unintended location within the DNA, producing changes that could cause disease or other harms. While some researchers indicates error may be rare, researchers do not fully understand the consequences of such errors. Additionally, CRISPR activity may stress cell and prevent editing. Further, while some cells can recover after their DNA has been altered, others cannot. Ethical concerns include whether CRISPR would be used for enhanced human characteristics such as increased muscle mass, learning aptitude and memory, equitable access to all populations, the potential to use CRISPR to edit the human germline like eggs, sperm and very early embryos also raise ethical questions. For example, to what extent might genetic changes affect future generations, and should research on human embryos be allowed? Another ethical consideration is the issue of equity and access. The availability and affordability of genome editing techniques could create inequalities in access to healthcare and agriculture advancement. It is crucial to ensure that genome editing technologies are used responsibly and ethically within consideration for fairness and social implications. Regulatory challenge. Many countries are struggling with the questions of how to regulate CRISPR and other gene editing technologies. For example, in the United States, it is currently unclear whether gene edited crops will be subject to the same regulation as conventional genetically modified organisms. In 2017, FDA began seeking public inputs on its regulatory policy, but as of March 2020, it has not published new guideline for industry on its current regulatory policy. In Europe 2018, a court ruling determined that gene editing crop would be subject to the same regulation as GM plants. The germline editing. Ability to edit DNA of reproduced cells raise ethical concern as any modification would be passed on the future generations. Off target effects. Genome editing techniques can have unintended consequences such as unintentional alteration in non-target genes. Human enhancement, like the potential for genome editing to enhance human traits, raise questions about it should be used for non-therapeutic purpose. Informed consent. The use of genome editing in research and therapy requires ensuring informed consent and transparency in its applications. Let us see certain future directions that are associated in genome editing. Genome editing has a potential to revolutionize the field of medicine. It offers promising possibilities for treating genetic disorders such as cystic fibrosis, sickle cell anemia and Huntington diseases. By editing DNA of affected individuals, scientists aim to correct and eliminate the disease causing mutations, offering hope for a future where these conditions can be effectively cured. Additionally, genome editing allows researchers to study the function of specific genes, leading to a better understanding of disease and development of a target therapy. It has also shown great potential for improving agriculture. By editing the genes of crop and livestock, scientists can develop crops that are resistant to pest, disease or environmental stress. This can lead to increased yield, reduced use of pesticide and improve food security. Additionally, genome editing can potentially eliminate allergenic protein from the food product, making them safer for consumption. The application of genome editing in agriculture has a potential to address global challenges such as food security. One of the main concerns is the potential for misuse or unintended consequences. The ability to edit the human germline, for example, raises questions about the ethics of altering future generations' DNA. There are also concerns about the potential for creating genetically modified organisms that could have unintended ecological impact. These are the certain lists where you can check out the further details of the related topic. I thank everyone for your kind attention. Hope this lecture has been fruitful for you. In the next lecture session, you will be able to learn about CRISPR-Cas technology in more detail. We will meet you in the next lecture session very soon. Till then, thank you.